Yeah, so our course project was based on a seed paper that analyzed epigenetic changes following a flu vaccine. So the main question was about why there's such a divergence in the efficacy of flu vaccines in comparison to modern vaccines we have. Because flu vaccines happen to be much less effective with rates around 40 to 60%. And the thing with vaccine is that they don't work by changing your DNA sequence, so the effect is achieved through epigenetic changes. And the main one we looked at was the modifications of histones. So the data set we had was called EPITOPH. It's a new modality that measures the abundance of 40 to 60 individual histone modifications at a single cell residue. And we had this data set at different time points before and after the flu vaccination, so we could get a sense of what are the most significant changes in histone modifications in immune cells, so that we could understand what works in people who have positive response to vaccine and what doesn't work.
My course project was this disease called polycystic ovarian syndrome, which is a really common disorder affecting anywhere between 4 to 20% of women all over the world. Although it's classified as a reproductive disease, it really is not. It's actually a metabolic disorder, and the problem is that it often gets treated only as a fertility issue, which leads to a lot of professionals having very specific opinions on how to treat it. However, the actual truth is that symptoms vary from one woman to another, and that's where precision medicine can really come in to help. My course project was about gathering data from 12 real patients suffering from PCOS and identifying which genes were upregulated and downregulated. My teammate and I managed to do that. And then we compared it to another data set which revealed which genes are more important for PCOS. We joined these two data sets and we found out which of these genes were actually important in disease regulation, and we figured out clusters for each of these genes, which will be really important in finding the gene ontology for these genes affecting PCOS.
In my course project, I worked with someone who has more of a biology background, and I came with a technical background. We were working on sort of just like analyzing cancer data sets. And we had like sort of two core data sets we were looking at. We were looking at one genomic data set and then one clinical data set. And the genomic data set to me was just really new. Like I've not really done in-depth analysis with the genomic data set before. So our project was like running this genomic data set through some of the pipelines that we had learned about in the class. And then on the clinical side, I think that was more of actually applying a lot of my pre-existing skills, because it was like a structured data set with different clinical indicators for people with and without cancer. So some of their demographic information and some of their lifestyle information. And so for this part, I was able to kind of train a standard machine learning model the way that I've done in the past. But on the genomic side, that was a lot more interesting to me in terms of like what I was learning, because I got to experiment with a new data set. The person I was working with had a ton of like really interesting insights that they brought to the table because of their domain knowledge. And we kind of were able to leverage that to just get some, interesting results.
My course project is using single-cell sequencing data and proteomic data to analyze Alzheimer's disease. The key part is to set up the infrastructure to scale the single-cell and proteomic data. And as you probably know, the single-cell transcriptome data come from every single cell of the patient lesion. And this is a giant amount of data. We were able to set up an infrastructure to actually have both analyses, the pre-processing process and the downstream analytic process.
Our project focused on using transcriptomic and metabolomic data from both pre-diabetic and diabetic patients, and looking at that data over time to see if we could identify transcriptomic or metabolomic determinants of diabetes onset. That was the idea behind it. As I mentioned earlier, actually working with the data and trying to run these pipelines was eye-opening. I think it all looked great on paper, but when we were actually we had issues accessing the data, had issues even running our preliminary analyses on subsets of the data, and then scaling it proved really hard, particularly because of the size of the data and access issues with accessing the data. So although we certainly didn't get to the endpoint that we wanted to, I think the project was really helpful in troubleshooting all those different steps. I think the most important thing that I took away from it was how to integrate data from outside sources into your own cloud computing pipelines. And even though that's not easy, I think the project really helped us in going through that process and troubleshooting different aspects of it.
Our course project was about organoids, which are basically artificial tissues that are created in a lab. And we looked at specifically pancreatic organoids. And we used basically like transcriptomic data to compare organoids of like pancreatic cancer with human cancer to determine if these organoids were reliable. And while we didn't implement the full thing, because it's a very complicated pipeline, we learned a lot about how to do scalability tests and basically learned about different statistical tests, differential gene expression that you can use to analyze data.
So my course project was looking at a radiomic approach to improve non-small cell lung cancer prognostication. So obviously small cell or non-small cell lung cancer is a very common disease in the States and globally. And one of the challenges with prognostication right now is that there aren't really great prognostication factors that go beyond just clinical factors such as age or performance status, etc. And so what I really enjoyed about this project was I got to work with imaging data, which I didn't have as much experience before. And I know that the course revolved mostly around genomic data, but because because the team that I formed was so interdisciplinary with someone was in statistics, another person did a lot of work with imaging data, I was able to not only learn about how to run pipelines from start to finish and learn how to incorporate that into Google Cloud, but also learn what the process of working with statistics and working with imaging data can look like from my peers as well as my teaching team. So I really enjoyed that kind of collaborative aspect of it.